Hey everybody. This video is going to show you how to extract DNA from a strawberry. Did you even know that you could do such a thing? Did you even know that strawberries have DNA? We're going to use a strawberry today. So this video actually is serving a couple of different populations. Number one, my students are doing this lab in class. And so this is a little how-to video for them, but I'm also putting this out on my social media because this is a super easy lab that you can do at home that's just kind of a wow factor for kiddos. And you probably have most of the items that you need to do this sitting around your house. And so you obviously are going to need a strawberry. You are going to need a plastic baggie. We're going to put the strawberry in there. You're going to need, this is the little lab kit that I hand out in my class. So my class has all the things that they need. You're gonna need some rubbing alcohol, isopropyl alcohol. So we have ours in a little tube just like this. You need some extraction solution. And I know that sounds very fancy. Right here, here's our extraction solution. This is made up of 9 parts water to 1 part dish soap, and then a little sprinkle of salt, just table salt like that you put on your French fries, sodium chloride. What this helps to do, we're going to pour it in with our strawberry in a minute, and it's going to break up the cells, because if you remember from your biology classes, the DNA lives in the nucleus of the cell, and so we want to break open those cells, and this extraction solution helps us do that. So you just mix 9 parts water to 1 part dish soap, whatever kind of dish soap you have laying around. That's our extraction solution. You are going to need some kind of funnel to kind of this is a little bitty tiny one my class is going to use. If you have a larger one in your kitchen, that would be fine too. You're going to need some kind of coffee filter or filter paper. In my class, yours may look like this. It might look like this, a little cupcake liner that's actually a teeny tiny coffee filter. Or it might look like this. So some of you have, I just used what I had around in the storage closet. So some of you got different kinds of filters. I'm going to use this one today. It tends to contain the mess a little bit better. So you need a filter. You don't have to have this next piece, but it's nice if you want to hold your DNA at the end. If you have a little jar, it's just going to come out in a little bitty thing. I give my students a little Eppendorf tube, and it just looks kind of like that. We're going to snap the lid shut at the end, and then you can carry your DNA around in there. So that is not necessary. The last item is a bamboo skewer. So we're going to use this to swirl out the DNA at the very end. So if you're ready, I'm going to walk you through this process and show you how easy it is to extract DNA from the strawberry. If you have the opportunity to, the rubbing alcohol should go in the freezer. If it's chilled before you do this experiment, it will work a lot better. Mine is not today, and you'll see it'll work just fine, but the colder your rubbing alcohol is, the better your results will be. So just if you have that opportunity, chill that in the freezer. All right, then we're going to take, you don't have to do this either, we're going to take off the stem, the cap of the strawberry. I'm just going to pull off. I have a giant strawberry here to use. I'm just going to pull the stem off. We're going to put it in, and we're going to pour that extraction solution. I have about 20 milliliters of this. You can eyeball it. It does not have to be perfectly measured. I'm going to pour that in with our strawberry, and then we're going to seal up this baggie. We're going to try to get out as much air as possible. So if you just kind of squeeze it all together and get out as much air as you can you see that? I'm just trying to squeeze out the air from the bag. All right, now the fun part. We've got our bag in, our strawberry in our baggie with our extraction solution. Now we get to mash the strawberry to smithereens. So we just take it and, oh, it's so fun. This is like therapy, a little stress reduction. So you just want to mash your strawberry. We're breaking open all of the cells, if you remember that we want to get to the DNA. So every cell in the strawberry has DNA. And we want to break those cells open. So you want to mush it as small as you can, being careful not to puncture the bag. Squeeze. You can probably do this for 3 minutes or so. I'm not going to quite do it that long. But just mash all of those pieces. Squeeze. breaking open those cells, getting that extraction solution down into all of those cellular parts that we want. All right, then we want to filter off all the solid pieces and just have the juice left behind. So that's what we're going to use our funnel and our coffee filter for. I'm going to need to point the camera down just a little bit. I'm going to use my little handy biology coffee mug here. Let me see if I can point this camera down just a little bit more so you can see. Well, I don't know if that helps. All right, so I'm going to filter it right back into my extraction solution tube. I'm going to put the funnel right in the top of that. It would be helpful if that had a flat bottom, but I did not purchase the kind that did. You can do this in any kind of material, just a jar, whatever you've got, okay? So I'm gonna use, this is gonna be a little awkward, but remember, we're just trying to get all the juice out. So we're gonna go kind of small. We're gonna do this in little stages. So we're gonna pour the mixture into our little funnel. Just very slowly and carefully, because it's going to make a mess. But we want the liquid to drip down through the funnel into so I'm going to go kind of slow and let that drip, drip, drip. Just get the liquid down in. Okay. So I'm probably about to make a huge, ginormous mess. But let's see if I can do this with a filter. All right. So you want to, whoops, see, science is messy. It's okay. You want to squeeze as much of that down in through the funnel, through the filter, so you're getting all the little strawberry juice without all the solid pieces. This little tiny filter is kind of silly, but it's what we had. All right, I'm gonna call that good, and I really wish I had some paper towels here. Woo, making a mess. We love science, we love making messes. I'm using a paper bag to wipe my hands because I don't have any other paper towels. So I have strawberry goopy hands. But we got what we wanted. Okay, let me take this label off so you can see it right there. So do you see the liquid that we have down in there? Okay, we've got our extraction liquid. So now what we want to do is pull out our isopropanol. This is our rubbing alcohol. Over the counter is fine. The higher percentage of alcohol it has, the better. But we're going to pour this very gently down the side of the tube. I have a lot of bubbles in here that I want to kind of stir down. All right, so here we go. I'm going to pour this gently down the side. You don't want to shake it up. Oh, yeah. All right, so I don't know if you can see this on my camera. Can you see those stringy bits in this layer right here? See the stringy bits? That's the DNA. I don't know how to get this focused a little better, but you can see it. All that little stringy stuff is the DNA. So we got it. We got it. Now, what you can do is kind of spool it out using your skewer. I'm going to swirl it up. It just looks kind of snotty. Oh, look at that. There's our DNA from our strawberry. And then what we like to do with it, for my students, they're going to put it in their Eppendorf tube, just like this. Just kind of wipe it off on the corner. Close the lid. And then my students are submitting a selfie with their DNA from their strawberry. So all of that mess to get this teeny little bit of DNA from a strawberry. If you would like the lab sheet from me, if you're a homeschooler or a science teacher or just a mom who needs something to do over spring break or on a rainy day, send me a message, send me an email, and I will shoot you this is the lab sheet that I use for my students. Now this is a college level class, but you know, anybody can read these and follow the directions. It even explains on the back how it works and how we get in and break open the cells and where the DNA is held. So I just think that's kind of a fun lab, and I hope you enjoyed it. And to my students, your lab is due on the Monday after spring break. If you have any questions, send me an email. Thanks, guys.